Hello, fellow warriors. Here we are with news from the website prostatewarriors.com. What if the key to breaking down an aggressive, seemingly unkillable prostate tumor isn't some billion-dollar, unpronounceable experimental chemical, but like a cheap, generic blood pressure pill sitting in millions of medicine cabinets right now?
Right. Or what if, instead of flooding the entire body with chemotherapy, we could just inject microscopic gold into a tumor and literally heat it up from the inside out?
I mean, those sound completely like concepts from a science fiction novel, right?
They really do. But they are active, real areas of research happening right now. The landscape of prostate cancer treatment is just shifting at a breakneck pace.
Yeah, it really is. And for anyone navigating this, whether you are, reading a pathology report for yourself or trying to understand the options for a loved one, the sheer volume of data can honestly feel like a tidal wave.
Absolutely. Which is why our mission for this deep dive today is to take a massive stack of the absolute latest clinical trials, cut through all that heavy medical jargon, and uncover the actual mechanics of how these new therapies work.
Spot on. And we are taking a very specific route to do this today. We're going to start on the clinical front lines, covering treatments already being tested in humans. We'll begin with phase 2 trials, then move downstream into phase 1/2 trials, and then phase 1 trials.
Right, where scientists are testing the absolute newest, cutting-edge ideas.
Exactly. And then finally, we will step out of the clinic entirely and head into the preclinical laboratories to look at the nanotechnology being built for the future. So let's just jump straight into the clinic.
Sounds good.
Our first stop focuses on optimizing the systemic therapies we already rely on, right? Like hormone blockers and chemotherapy.
It makes perfect sense to start there, because before we invent entirely new weapons, we really need to know if we are using our current arsenal to its maximum potential.
Right. Which brings us to a really compelling phase 2 study called the ARASEC trial. This one looked at patients with metastatic hormone-sensitive prostate cancer.
Yeah, and the numbers on this one just jump right off the page. They combined standard androgen deprivation therapy, or ADT, with a hormone-blocking drug called darolutamide.
And the results were massive, right? The patients getting this doublet therapy saw a 71% reduction in the risk of disease progression or death.
A huge reduction, yeah.
But looking closely at how they actually ran this trial, I'm a bit confused by the setup. Usually, a clinical trial has two groups right there in the clinic. The group getting the new combo and a control group getting the old standard treatment. But ARASEC didn't have a traditional control group.
No, it didn't.
Mm-hmm.
And you are hitting on a major logistical hurdle in modern trial design. Medical guidelines just move so fast now. Giving a control group standard ADT alone is currently recognized as insufficient care for this stage of the disease.
Oh, I see. So ethically, you can't really do that.
Exactly. You simply cannot enroll a patient in a modern trial and intentionally give them an outdated substandard treatment just to prove a point.
Okay, that makes total sense. But if you can't have a real-time control group, how on earth do you prove your 71% reduction is actually scientifically valid?
Well, the researchers use this really fascinating statistical technique called propensity score matching.
Propensity score matching. Okay, what does that actually mean?
Basically, they took the 223 patients actively participating in the ARASEC trial, all of whom received the intensified darolutamide combination. Then, they dug into the historical data from a massive, much older study called the CHARTED trial.
Oh, wow. So they use old data.
Yeah, they combed through that old data to basically build a perfectly matched ghost control group.
Wait, a ghost control group, when you say perfectly matched, how precise are we actually talking here?
Oh, incredibly precise. They aligned these historical patients across 6 crucial metrics. They match for age, they match for PSA levels, and they match for a Gleason score.
And Gleason's score is basically a measure of how aggressive and mutated the cancer cells look under a microscope.
Exactly. And they even match for performance status, which is it's a clinical scale rating how well a patient can carry out normal daily activities without fatigue. So for every single real-world patient in the ARASEC trial, the algorithm found their historical twin who had only received the standard ADT.
That is just a brilliant workaround. You get rigorous comparative data without ever having to compromise the active care of a single person in the clinic.
It really is. And it solidly proves that for patients who maybe can't tolerate aggressive chemotherapy, this dual hormone approach is incredibly powerful.
But speaking of chemotherapy, let's look at another trial that tried to combine our heavy hitters. This was a phase 1/2 trial testing enzalutamide, which is a very potent hormone therapy given at the exact same time as Cabazitaxel, a serious chemotherapy drug.
Right, and they reported some very deep biochemical responses initially. Over 61% of patients saw their PSA drop by 90%.
Which sounds amazing. But wait a second. If you hit a patient with an intensive hormone blocker and a toxic chemo simultaneously, aren't you just completely overwhelming their liver? Like, does throwing everything at the wall at once actually work biologically?
That skepticism is entirely justified, honestly, and the trial results actually back up your concern. While those initial PSA drops looked great on paper, the researchers uncovered a significant drug-drug interaction when they looked at the pharmacokinetics.
Okay, and pharmacokinetics is essentially the study of your body's plumbing system, right?
Yeah,
How a drug is absorbed, broken down by your liver, and cleared out.
spot on. It's the plumbing system.
So what exactly happened when the two drugs entered that plumbing system together?
Well, the enzalutamide actually sabotaged the chemotherapy. You see, enzalutamide is known to ramp up the activity of certain enzymes in the liver.
Uh-huh.
When given concurrently, those hyperactive enzymes just chewed through the cabazitaxel way faster than normal. It dramatically reduced the actual exposure of the chemotherapy drug circulating in the patient's body.
So wait, the hormone therapy was essentially digesting the chemotherapy before it could even do its job.
Precisely. It highlights this critical lesson in oncology, which is that timing and sequencing are just as vital as the active ingredients themselves.
That is wild. I guess you can have the two best drugs in the world, but if they are literally fighting each other in the liver, the patient loses out. It really proves why we can't just blindly stack systemic treatments that flood the entire body.
Exactly.
So if flooding the entire bloodstream creates these complicated plumbing conflicts, the natural next question is, can we just bypass the whole system and drop a bomb directly on the tumor itself?
Right. And that is exactly where the field of next-generation radiation is heading. We're moving away from whole-body drugs to targeted radioligands, smart bombs that seek out specific proteins on the cancer cell.
Yes, and we have a prime example of this in a phase 1 trial testing a next-generation PSMA targeted radioligand. It goes by the name 177 Lu-LNC Quan 11. They tested this in heavily pretreated patients, and at the highest dose level, 66.7% of them achieved at least a 50% decline in their PSA.
Which is very significant for heavily pretreated disease.
Utterly. To give you a mental picture of why this specific drug is a big deal, think of existing first-generation radioligands like a tourist. They enter your bloodstream, they visit the tumor to deliver a quick burst of radiation, and then they're flushed out of the body pretty rapidly. But 177 Lu-LNC 1011 has a chemical tweak called a Dancel modification. It doesn't act like a tourist, it acts like a grappling hook.
That's a great analogy. The Dancel modification is a structural change that radically alters how long the drug actually survives. Current therapies like Pluvicto do a great job, but they clear through the patient's system relatively fast. This new modification gives 177 Lu-LNC 1011 a 128-hour effective half-life inside the actual tumor.
Wow, 128 hours. Let's translate that into the daily reality of a patient. What does that actually mean for the person sitting in the treatment chair?
It means the drug drops anchor and stays bound to the cancer cell for over 5 days.
Oh, wow.
Right. For the patient, that translates to a massive, sustained dose of radiation localized exactly where it needs to be, all while they're at home, eating dinner or sleeping. It maximizes the damage to the tumor while minimizing the radiation circulating through healthy tissue.
That's incredible. And speaking of maximizing damage, there is another phase 1/2 trial doing something similar called the Prospector trial. They're combining a targeted alpha therapy called AZD2265 with a drug called AZD9574, which is a PRP1 inhibitor.
Yeah, this combination relies on a really cool biological concept called synthetic lethality. Think of it as a 1-2 punch where neither punch is fatal on its own, but together they are devastating.
Okay, break that down for me.
Sure. So the targeted alpha therapy, the AZD2265, acts like a microscopic sledgehammer. It delivers high-energy alpha particles that physically shatter the DNA strands inside the prostate cancer cell.
Ugh.
Now, normally, a cancer cell won't just die from that. It will call in an emergency repair crew to stitch its DNA back together.
And that repair crew is the enzyme called PARP1. So the alpha therapy shatters the DNA, and then the AZD9574 sneaks in and essentially steals the cancer cell's toolkit.
You've got the mechanism down perfectly. The alpha therapy creates the catastrophic damage, and the PRP inhibitor ensures the cancer cell cannot fix it, which forces the cell to die. What makes this specific PRP inhibitor, AZD9574, so important is its selectivity. It is 8,000 times more selective for the PRP1 enzyme than other related enzymes in the body.
Okay, 8,000 times. Why does that specific figure matter so much to the patient?
Because precision limits collateral damage. Older drugs that block multiple enzymes often interfere with healthy cells,
Hmm.
and that causes severe nausea, fatigue, or major drops in blood counts. By being 8,000 times more selective, this drug ideally just steals the toolbox from the cancer cells, leaving the patient's healthy tissues alone.
That is fantastic. But there is a massive catch to all of these PSMA-targeted therapies we've discussed so far. Prostate cancer is notoriously adaptable. Eventually, the cancer figures out it's being targeted, and it simply stops expressing the PSMA protein. The target disappears, and the cancer hides.
Yeah, it's a major, major resistance pathway. When the tumor stops showing PSMA on its surface, all those smart bombs that we just talked about suddenly become totally blind.
Which makes this phase 1 compassionate use trial for a drug called ARASEC Dodone so critical. Instead of looking for PSMA, this drug targets a completely different protein on the surface of the cell called ACP3.
Exactly. Using a radioactive isotope called lutetium-177, they treated a man whose cancer was heavily pretreated and actively progressing. Once they switched the target to ACP3, he experienced a PSA decline that lasted for over 200 days.
That's huge. Now, finding a new target is obviously great for survival, but targeting ACP3 specifically solves a miserable side effect issue too,
And
right?
it addresses a massive quality of life issue, yes. Standard PSMA therapies have this notorious flaw. The PSMA protein is also naturally found in your salivary glands and your lacrimal glands, which produce tears.
Oh, right.
So when you take a PSMA radioligand, radiation accumulates in those glands. Patients suffer from severe dry mouth and dry eyes. And it's not just a minor annoyance, it can lead to rotting teeth, difficulty swallowing food, and constant eye pain.
It sounds awful, but ACP3 avoids that completely.
It does. ACP3 is highly specific to prostate tissue and is almost non-existent in the salivary and lacrimal glands. By switching the crosshairs from PSMA to ACP3, the drug bypasses those glands entirely. The tumor gets destroyed, and the patient gets to keep their saliva and their tears.
That is a phenomenal upgrade for everyday life. Okay, so radiation is essentially destroying the tumor from the outside in, but let's look at how we can reprogram the body to fight from the inside out. We are seeing incredible momentum in harnessing the immune system right now. There's a phase 1 trial testing PSMA-targeted CAR-NK cells. They tested this in 8 heavily pretreated men, and half of them saw a PSA decline of over 30%. Now, we've heard of CAR T cells before, but CAR-NK is different, right?
Very different. CAR T-cell therapy is famous for its success in blood cancers, but it is an agonizingly slow, custom-built process.
How so?
Well, you have to extract the patient's T-cells, physically ship them to a specialized laboratory, genetically engineer them to hunt cancer, and then ship them back to infuse into the patient. It takes weeks, costs an absolute fortune, and the patient just has to wait.
Wow. Whereas these CAR-NK cells, natural killer cells, are described as off the shelf. They are pre-engineered from healthy donors, non-viral, and literally just sitting in a freezer ready to go the moment a patient needs them.
Exactly. They act as a rapid strike force. But the real advantage actually lies in how they behave once inside the body. See, CRT cells are designed to persist. They multiply rapidly inside the patient.
Which sounds good, right? More cells to fight the cancer.
It sounds good, but it often triggers a terrifying immune overreaction called a cytokine storm.
I've heard of cytokine storms. That's when the immune system basically panics, catches fire, and starts attacking healthy organs, right?
That's exactly it. The immune system releases a flood of inflammatory proteins, causing extreme fevers, plummeting blood pressure, and sometimes total organ failure. But because natural killer cells don't rapidly multiply and persist in the body the same way T cells do, they just do their job, they assassinate the cancer cell, and then they naturally clear out. The risk of those life-threatening cytokine storms is drastically reduced.
We're seeing a similar effort to make treatments less taxing on the patient in another phase 1 trial for a drug called GTB-5550. This is also an NK cell engager, but it seeks out a different target called B7H3. What stands out to me here isn't just the biology, but the delivery. It is a subcutaneous injection, literally just a quick shot under the skin, rather than chaining a patient to an IV drip for hours in an infusion center.
Reducing the friction of treatment is a major, major theme in these early-stage trials. But as we look at these phase 1 and phase 1/2 trials, we also have to talk about the mystery molecule.
Oh, this part is wild. There's a phase 1 trial for a drug called INR-731 and a phase 1/2 trial for a drug called GDC-1261. GDC-1261 has 260 patients enrolled in it. And yet the pharmaceutical developers have kept the exact biological targets a complete secret from the public.
It's true.
I just have to express some genuine disbelief here. How does a massive 260-person human trial get approved and run without the broader medical community knowing what the drug is actually attacking?
I know, it seems totally counterintuitive to the collaborative spirit of science. But oncology is a fiercely competitive, multi-billion-dollar industry. When a developer sees a remarkably strong biological signal in the lab, they will rush it into clinical trials while keeping the exact mechanism proprietary so competitors can't steal the concept.
Right.
However, they usually leave a trail of breadcrumbs.
Like the clues in INR 731 trial.
Right. INR 731 is an oral pill. And if you look closely at the trial's inclusion criteria, they specifically exclude patients who have previously been treated with cereblon-dependent degrader therapies.
Oh, interesting.
That incredibly specific exclusion totally gives the game away. It strongly implies the drug works by hijacking a protein called cereblon, which is part of a cell's natural garbage disposal system. The drug basically tricks the cancer cell into tagging its own survival proteins as trash and grinding them up.
It's molecular espionage. Okay, so we've looked at new immune cells and secret drugs. But there's another approach happening right now, taking familiar, older drugs and using them in completely new ways to defeat the cancer's resistance. There is a phase 1b trial out of Roswell Park combining enzalutamide with a drug called venetoclax, which has been FDA approved for years to treat blood cancers.
Yeah, this trial is a beautiful example of anticipating the cancer's evolutionary next move. We know that when you give a patient enzalutamide, you are blocking the hormones that feed the cancer. The tumor begins to starve. Biologically, the cancer cell panics.
Right. It's trying to survive.
Exactly. So in a desperate bid for survival, it raises a microscopic shield, a protein called BCL2. This shield physically prevents the cell from dying, keeping it alive just long enough to mutate into a more aggressive, resistant form.
So the researchers bring in venetoclax because venetoclax specifically hunts and destroys that BCL2 shield.
You got it. You anticipate that the cancer will try to raise the shield, so you give venetoclax simultaneously to rip it away. You make the tumor vulnerable again before it ever has a chance to adapt.
And speaking of unexpected older drugs, we have a phase 1 trial out of Dartmouth using Telmisartan. For those unaware, Telmisartan is an incredibly common generic blood pressure pill. Yet they are testing it against advanced prostate cancer in combination with PARP inhibitors.
It's fascinating.
Right. A blood pressure pill fighting cancer sounds like a typo. How does this actually work?
It all comes down to the environment surrounding the tumor. Solid prostate tumors are messy. They grow these chaotic, leaky, and disorganized blood vessels. Because the blood supply is so poor, the center of the tumor becomes deeply hypoxic, meaning it lacks oxygen.
And hypoxia is a bad thing for treatment, right?
A very bad thing. Hypoxia is a major driver of resistance. Without oxygen, radiation and PRP inhibitors simply do not work effectively.
So the telmisartan comes in and acts on the blood vessels.
Yes. Telmisartan helps dilate and normalize those chaotic blood vessels. It basically restores proper blood flow into the deep tissues of the tumor. Suddenly, the hypoxic zones are flooded with oxygen.
Oh, wow.
And not only does this allow the chemotherapy to physically penetrate deeper into the mass, but the presence of oxygen acts as a catalyst, making the DNA damage caused by the PARP inhibitors permanent and lethal.
It essentially alters the battlefield so our standard weapons hit harder. That is incredible. And that brings us to our final stop today. We are leaving the clinic, stepping away from human trials, and looking at the preclinical horizon. This is the laboratory research focusing on nanotechnology. How do we build microscopic vehicles to deliver these drugs flawlessly?
It's so important because delivery is the silent killer of great drugs. If a compound can't physically cross the membrane of a cancer cell, it is useless, no matter how potent it is in a petri dish.
Which makes this project using Orlistat at CSRI Nanoparticles so fascinating. Orlistat is an over-the-counter weight loss pill, but researchers discovered it also happens to block an enzyme that prostate cancer cells desperately need to synthesize their own fat. To get it inside the tumor, they packed the Orlistat into microscopic spheres called Cis-8e nanoparticles.
The mechanism of destruction here is terrifying for the cancer cell, honestly. When the nanoparticle drops the Orlistat inside, it forces the cancer cell to radically change its own structural fat composition. It fills the cell membrane with highly unstable fats called PUFAs, polyunsaturated fatty acids.
And why are PUFAs unstable?
Because they are highly reactive to oxygen stress. It is the cellular equivalent of taking a stone fortress and secretly replacing all the bricks with fragile glass. The moment the cancer cell experiences normal metabolic stress, those PUFA fats undergo lipid peroxidation. The cell membrane literally rusts, fractures, and the entire cell falls apart.
That is an incredibly elegant way to destroy a cell. But, if you want a true glimpse into the future, look at this preclinical study out of the National University of Singapore. They are building DNA barcoded gold nanoparticles.
This one is amazing.
It really is. Their goal is to deliver genetic therapies straight into the mitochondria, the power plant of the cancer cell. Once the gold is inside the power plant, they shine an infrared light on the tumor. The gold absorbs the light, heats up rapidly, and incinerates the power plant, resulting in 99% tumor regression in their models.
Yeah, the therapeutic heat is impressive. But the true revolution here is how they discovered the correct nanoparticle shape in the first place. Historically, finding the right nanoparticle to penetrate the mitochondria was just a game of trial and error. You build one design, inject it into an animal model, wait weeks, and see if it works. It takes years and thousands of animals.
Right. But these researchers didn't do it one by one. They built 30 different microscopic designs at the exact same time. And to keep track of them, they attached a tiny, unique sequence of DNA to each one, basically a barcode. It's like attaching a Jeep Zest tracker to 30 different microscopic delivery trucks.
Exactly.
They injected all 30 variations simultaneously. Then, they simply harvested the mitochondria and read the DNA barcodes to see which specific truck successfully parked inside.
The efficiency of that approach just cannot be overstated. By testing 30 designs at once, they generated over 1,000 data points on how shape, size, and surface chemistry affect cellular penetration. And they accomplished all of this while using 30 times fewer animals than traditional methods. It condenses years of tedious laboratory discovery into a single, high-throughput experiment.
It's brilliant. We have covered a massive amount of ground today. From ghost control groups bypassing ethical dilemmas to anchors that hold radiation in the body for 128 hours, to repurposing blood pressure pills and weight loss drugs. For you listening right now, the relevance of all this complexity boils down to one very reassuring truth. The medical community is no longer looking for one singular, elusive magic bullet. That era is totally gone. Instead, they are waging a coordinated, multi-front war. They are optimizing your hormones, hyper-targeting radiation, programming off-the-shelf immune strike forces, and building microscopic golden vehicles. The sheer volume of simultaneous innovation is staggering.
It's a truly remarkable time in oncology, and it leaves us with an intriguing question to mull over. As we start relying on DNA barcodes and artificial intelligence to test 30 different microscopic nanobots simultaneously in the lab, is the future of cancer treatment going to be less about discovering brand new exotic chemical drugs and more about perfecting the microscopic vehicles we use to deliver the tools we already have?
Stay strong and fight on.